My name is Michelle Jabor, and today's date is 5/19 of 2022. In today's presentation, I'll be discussing the anatomy and the physiology of the human retina. The human retina is defined as the innermost layer that coats the innermost layer of the human eye. The retina itself is subdivided in two different categories, which could be divided into two, as shown in this image here. We have the first layer, that is the retinal pigmented epithelium, and then we have the neural retina. The first category that we will be discussing today is the retinal pigmented epithelium. This is the outermost layer of the retina, as it's observed down here in this image, and it is formed of cuboidal epithelial cell, of a single layer of cuboidal epithelial cells, and as mentioned, they are the outermost layer of the retina. This layer has different responsibility in the human retinal cells. They do give support and nourishment to the neural retina in the first place, and then they play a role in the visual cycle as they do regenerate the photosensitive pigments. Inside the retina, there are different cells that exist. They could be divided into three different categories. The first one being the photoreceptors, the second one is the neural cells, and the third one being the glial cells. To discuss these in a little bit more details, the first category of the cells is the photoreceptor cells. As the name say, they receive the light, which means that these cells are responsible to respond to the light. They're light sensitive, and then they convert the light that they get to a stimulus that will get to the brain through the optic nerve from the eye, so we can see and form an image. The second category would be the neural cells. These cells have five different types within them. They have the photoreceptors that are very important in the eyes, known as the rods and the cones, as well as the bipolar cells, the ganglion cells, the horizontal cells, and the amacrine cells. And then lastly, we have the glial cells, which are used in the retina to maintain the homeostasis of the eye. Now, the retina has so many different layers to it. Specifically, there are 10 different layers to the retina, which are listed and shown in this image right here. Going from the interior to the posterior, we have the inner limiting membrane, the nerve fiber layer, the ganglion cell layer, the inner plexiform layer, the inner nuclear layer, the middle limiting membrane, the outer plexiform layer, the outer nuclear layer, the external limiting layer, and most importantly, the very last layer that we observe is the layer that has the rods and the cones, which as mentioned before, one of the neural cells of the ganglion, or of the retina, The most important part of the retina that we're going to be focusing on today is going to be the neural cells that do exist in the retina. They are 6 different ones, and they each have a very specific role that they play in the reception of the light, in the processing of the light, and getting the light to the brain. First of all, we're going to start by the rods. These are the most abundant, one of the most abundant cells within the layers. There are about 120 million rods in the human retina. They are usually found on the periphery of the retina. Very little of them, or none of them, could be found in the central of the retina. These are very, very light sensitive. They are very light sensitive, and there are about 1,000 times more sensitive to the light than the cones are. They get their name from their shape. If you look at them, they kind of have this rod shape. Since they're very light sensitive, they're going to be responsible for the light vision. They're better in the dark, so we call them, they're responsible for dark vision, or what we call black and white vision. The next type of cells that we're going to be looking at are the cones. The cones are receptors just like the rods. They exist in less amount than the rods. Again, they get their name from their shape. They kind of look like a cone as well. The difference between these two is the location. We said that the rods are present on the periphery of the retina, whereas the cones are present more in central, in the middle of the retina. At one point, the retina has a dimple, it dimples, and that dimple is what we call the fovea. The fovea is responsible for us to be able to see more detailed images. The cones are located right in front of the fovea. That's where they are very, very concentrated. And then there are very minimal ones on the periphery of the retina. They are less color sensitive than the rods are, which make them better for color vision. Within the cones, there are three different types of subtypes of cones, to be said better. The first one is the red cones, which make up about 60% of the full amount. The second one would be the green cones, which make about 30%. And lastly, we have the blue cones that make up about 10%. As we mentioned, in the retina, the peripheries have the rods, and then the retina dimples, and we have the fovea, and then we have the cones right there. There is a spot in the retina where the fibers and these tissues and these cells exit the retina into what we call the optic nerve. Right in front of that optic nerve, there are no receptors at all. This is what we call the blind spot of a person, where there are no receptors at all. Third one that we're going to be talking about is the retinal ganglionic cell. It is a neuron type located in the inner surface of the ganglion cell layer of the retina that was mentioned before. It receives the visual information from two different neurons, one being the bipolar cell and the other one being the retinal amacrine cells. Then they will help these information go into the axon to be transported to the brain. The fourth type of cells that we're going to be talking about are the bipolar cells. These cells are interneurons which are present in the second layer of the retina, and then they transform visual information from the photoreceptors, so they get their information from the rods and the cones, and then they're the ones responsible to transfer them to the ganglion cells. They are the only neurons, or they are the only cell that connect the outside of the cell to the inside of the cell. The bipolar cells in the eye are one extra layer of cells that are usually not present in any other type of cells. Next, we have the horizontal cells. These are laterally interconnecting neurons having cell bodies in the inner nuclear layer of the retina that was mentioned before. They are responsible of modulating information transfer between the bipolar cells and the photoreceptors, and they help the eye adjust to both bright and dark or low light conditions. Lastly, we'll talk about the amacrine cells. These are interneurons that interact at the second synaptic level of the pathway that has the photoreceptors, the bipolar cells, and the ganglion cells. They are the major carriers of road signals to the ganglionic cells as well. Moving on, we're going to talk about what happens when the light hits the rods or the cones. So what happens in that retinal layer? Shown here are a rod and a cone. And as mentioned, they get their name from their shape. One is shaped like a cone, one is shaped as a rod. So after the light goes from the pupil to the retina, it's going to go to these photoreceptors that are the rods and the cones. Within the rods and the cones, and I'm going to be focusing on the cones, even though the shapes are different, but the inner structure is very similar. Both of these do have discs in them, a bunch of discs that are stacked on top of each other's. These discs have some proteins or they have some elements on them that are going to be responsible, or that are going to be photoreceptors. And these proteins are called rhodopsin in the rods and photopsin in the cones. Inside these proteins that are located on the disc is going to be little molecules that are called retinal. These retinal within the little photoreceptors are bent in shape, or they are called in an 11 cis shape. When the light hits and then gets to the rods and then to the rhodopsin, they get to the retinal. Once they get to the retinal, what happens is that this retinal will change its shape. And then after that, after changing its shape, it's going to go from being bent to being linear. This will initiate what is called a phototransduction cascade. This phototransduction cascade is basically related to what happens inside of the retina. Inside there is what's called a cyclic GMC. The cyclic GMC, as long as it's present and it's bound, it keeps the sodium channels inside the retina open, which keeps the flow of the retina, or which keeps the flow of the Na into the cell. Once the transduction cascade happens, there are what's called an alpha molecule that comes off of the surface and starts breaking the cyclic GMP into GMP. Once that level of cyclic GMP starts going down within the cells, the ionic channel of the sodium closes. Once these close, what happens is that there is no more sodium that can come into the cell, and the cell becomes hyperpolarized. So this is kind of what happens inside the molecule. At dark, when there is no light at all, the receptors inside the rods or the cones are activated, which means because the channels are open, because there's Na+ floating, there are depolarized, there is a depolarization, there is a resting member potential, there is a depolarization, and they're activated. The receptors are activated. When the receptors are activated, they will activate the next cell in line, which is the bipolar cell. The bipolar cell by nature is inhibitory. So its action is it inhibits whatever comes after. So what that means is that the bipolar cell will inhibit the ganglionic cells. The ganglionic cells cannot work, so they cannot send a signal to the optic nerve, which means that the brain don't see anything. There is no signal. We see dark. In the light, when the light hits the rods and the cones, to the rhodopsin, to the retinal changes its shape, the alpha molecule come off, it breaks down the cyclic GMP to regular GMP. A low concentration of cyclic GMP will let the ion, the Na+ ionic channel close. No more Na+ can come in. We have a hyperpolarization. When we have a hyperpolarization, the receptors are shut off. When they're shut off, or when they're deactivated, they cannot activate the next cell in line, which is the bipolar cells. The bipolar cells are now inactive. They cannot work. So this inhibitory action is inhibited. So we negated a negative, which means that now they're not working. The ganglionic cells are now active, and then they can send a signal to the nerve, to the optic nerve, which sends it to the brain, and we see light. So this is the structure and the physiology of the retina. Thank you.